So today we're going to talk a little bit about the chemical reactions necessary to sustain life. And collectively they're known as metabolism. And metabolism can be broken down into two broad categories, anabolic and catabolic reactions. Now, anabolic reactions take small chemical components and attach them together to build larger molecules. And that's a type of chemical reaction that requires energy in order to proceed. So we call it an endergonic reaction. And then catabolic reactions, which take large molecules and tear them apart for the purpose of getting the energy out. Okay, and the energy that is immediately available to do work is called ATP, and the job of the catabolic reactions is to transfer the energy that's stolen from fuel molecules over to ATP so that we can use it to do work. And without energy, we can't do anything, which means we would die, right? So all of these things that are listed here, none of them would be possible if we didn't have energy, okay? We don't need captions. The energy that we get comes from the nutrients that we consume, and the main nutrients are proteins, carbohydrates, and lipids. We also consume vitamins and minerals, all right? And all of these are necessary in the production of energy, which is the ability to do work, right? Energy comes in a lot of different forms. Two broad categories of energy are potential energy, which is the energy of position, and kinetic energy, which is the energy of movement, okay? You can convert from one form of energy to another. In fact, you can demonstrate this by taking the elevator to the second floor of this building, right, and changing your position from the first to the second floor. And then if you wanted to convert that potential energy of position back to kinetic energy, all you'd have to do is fall out the window, okay? Where would the energy go? Well, some of the energy would be lost as pressure, some would be lost as sound, some would be lost as heat, right? But energy transfers happen all the time, and as we know from the second law of thermodynamics, they are not 100% efficient. They lead to heat going off into the universe, and that heat serves to make the universe more random, right? So we say in the second law of thermodynamics that the universe tends towards maximum disorder or entropy. Another way of stating that is that if we consider any system, that system will always arrange itself in a way that takes the least amount of energy to maintain unless you put energy into that system, okay? Classes of energy, well, we've got potential energy. An example in living systems is the potential energy in the plasma membrane, which is the result of concentration gradients that are created as a result of membrane pumps and differential permeability of the membrane to different ions, and the fact that certain large ions are trapped inside the cell and can't escape. And that means that there's a separation of charge across the membrane that is a form of stored energy, just like the charge in a battery, right? And we can change that potential energy into current by opening a doorway that lets the opposite charges run into each other, right? And that moving series of charged particles can be used to do work, such as transport, or in the case of nerve cells, send electrical signals, or in the case of muscle cells, contract, okay? There's also potential energy in the shells of electrons around the nucleus of an atom, right? We know that the outermost shell has the most energy, and that's where the bonding takes place. And then the shells that are closer to the nucleus have less energy, okay? Potential energy has to be converted to kinetic energy before it can do work. You can see some examples of these different forms of energy here, right? You can see potential energy across the membrane, with a laser pointer up here, okay? where you've got a higher positive charge on the outside than the inside. And then when we open a doorway to let that flow take place, then we have current, and that current can be used to do work, right? And in the atom, it's the position in the shells around the nucleus of an atom that determines the amount of energy you have as a result of your position, right? That can be used to do work as well. You can also see electrons changing their location as they move through something like the electron transport chain in the intermitochondrial membrane, and that energy gets trapped by the enzymes that are embedded in the membrane, and it'll be used to create the proton gradient across the inner membrane that can ultimately be used to do work. Okay? So all sorts of different energy transformations that take place, right? Chemical energy is a form of potential energy, energy that's in an electron's in a molecule's chemical bonds can be stolen by cracking that bond, and then we can use it to do other work by first transferring it to ATP. Okay? Molecules that are rich in this type of energy include our major nutrients, right? Triglycerides, which are lipids, and glucose, which is a carbohydrate. And of course, ATP has a lot of useful energy, primarily in the 3-phosphate groups that are attached to the molecule. And that's because they have a negative charge on them, so the covalent bonds that hold them together are very powerful. Okay? Electric energy is just the movement of charged particles. If you're talking about electricity flowing through a power cord, right, that's the movement of electrons. If you're talking about current in living systems, that can either be the movement of positively charged particles like cations, or it can be the movement of electrons, such as in the electron transport chain. And all of that can be captured to do work. Mechanical energy is just force applied to objects in order for them to move, right? And then sound energy is a pressure wave that moves through a medium, such as a liquid or a gas, in order to produce movement. Radiant energy, or electromagnetic radiation, is a form of energy that has both an electric and a magnetic component. They are at right angles to each other as the energy travels through space. And the electromagnetic energy can be used to do a variety of things. It can break chemical bonds. It can, in some cases, damage living tissue. It can be captured by plants in a process known as photosynthesis and used to make carbohydrates such as cellulose or wood and starch, right? So this energy is unusual in the sense that it's the only form that can move through a vacuum, right? The other forms of energy generally require a medium through which to move. Heat is an electromagnetic radiation. It is at the low end of the energy spectrum of electromagnetic radiation, and it generally is not available to do work, and it's usually expressed as the temperature of an object. The electromagnetic spectrum shown here indicates that the shorter the wavelength, the higher the energy content. And the reason for that is an equation. Here we go. Let's get the equation up here. The equation is E equals H nu. So let's write that. Okay. E equals H nu. Okay. Where nu is, this is frequency, this is a constant, and this is energy. And then the other equation you need to know is that 1 over the wavelength, which is expressed as lambda, is equal to the frequency. Okay? So what these two equations tell you when you take them together is that the shorter the wavelength, the higher the energy content, and vice versa. So the real dang stuff is way over here on the left, and the less energetic material is way over here on the right. And there's a visible light there, kind of in the middle, okay? More energetic than visible light, UV, X-rays. You know, you can't be around X-rays very long before you damage living tissue and cause mutations, right? And gamma rays are the same way, cosmic rays are the same way, right? But you don't have anything to fear really from infrared and radio waves because the energy content is so low, okay? The fact that there's an electrical and a magnetic component to this can be demonstrated if you've ever seen pictures of Las Vegas, Nevada, and note all the lights there. The energy that is used to generate all that electricity actually comes from a lake north of town called Lake Mead, which was created by the Hoover Dam, damming up the Colorado River. And what happened is that once the dam was built, they put turbines inside the dam, and then as the water flowed down the dam, right? So this would be this side view here. This would be the dam. We'll draw the dam here. Okay. Okay, and then we'll draw the water. Okay. Pen color blue. Right, there's the water on one side of the dam. That's Lake Mead, right? And then the water goes up and over the dam, and as it flows over the dam, the turbines inside the dam are like little wheels. Draw them in black, I guess. Okay. And then as the water turns the wheels, that's energy, potential energy, right? Energy of position, turning into kinetic energy of the falling water, turning into kinetic energy of the spinning turbine, right? The turbine has magnets on it, and in the core of the turbine is a copper coil, and it's the property of a moving magnetic field that you induce an electrical current in any nearby metal. And that's exactly how Las Vegas gets all its electricity, and that's called hydroelectric power, right? You've also seen this at the junkyard. If you've ever seen cars picked up and dropped in a crusher, that's an electromagnet, right? It's a big iron disc, and it's not a magnet until the operator cuts the current on, and when the current runs around the big iron disc, it induces a magnetic field in the disc, and he can use that to pick up the car. And then when he gets the car over the crusher and he cuts the current, the magnetic field disappears, and the car falls into a crusher and you smash it, right? This is an unusual kind of energy, right? And it's used in biological systems in the form of photosynthesis. It's also used in the human body when we convert light into electrical information in the retina of the eye, and that electrical information gets sent back to the occipital lobe of the brain, where we turn that into information about vision, okay? So all of this energy content, all of these forms of energy can basically transform into each other. The first and second law of thermodynamics we actually discussed during the chemistry chapter, right? The first law states that energy and matter can't be created or destroyed, but they can be converted into each other. And the equation for that is E equals mc squared, right? Where E is energy and m is mass, and c is the speed of light, right? So what it tells you is if you take a tiny amount of matter and you convert it into energy, you get a whole lot of energy coming out, right? And that's the equation that gave us the atomic bomb and atomic power, all right? And the second law of thermodynamics, which we've already discussed, which is that if you consider any system, all right, you don't put any energy into the system will try to get to the low energy state, okay? Any way it can do that, because it doesn't have the energy available to maintain a high energy state, right? A low energy state governs everything, right? So in metabolism, we're talking about all the chemical reactions that take place in order to sustain life, and we can break those down into anabolic and catabolic reactions, as we already discussed. Chemical reactions occur when chemical bonds in existing molecular structures are broken, and energy comes out, and then some of that energy can be used to make new chemical bonds. The reactants are the things on the left-hand side of the equation that produce the products on the right-hand side of the equation. Okay? So the reactants may bump into each other, or the reactants may split apart, right, and generate the products, right? The reactants are always, by convention, written on the left-hand side of the chemical reaction, and the products are always written over here on the right-hand side. These are the products, okay? In order for a chemical reaction to happen, movement has to occur, right? Generally, a chemical reaction involves the collision of two or more objects in order to generate a product, and that collision has energy associated with it that's used to make and break chemical bonds, right? Break old ones and make new ones, for instance. The direction of the reaction is indicated by an arrow. Sometimes you'll see a double arrow, which indicates that the reaction can go in either direction, and that indicates something known as an equilibrium reaction, okay? And we always obey the first law of thermodynamics, which says that in a balanced equation, the number of atoms of a particular type on the left side are equal to the number of atoms of that particular type on the right side, okay? So we always have to write a balanced equation in order for the reaction to be meaningful, okay? Chemical reactions can result in changes in chemical structure and energy, and can indicate whether the reaction is practically, in real terms, irreversible or reversible. The three main types of reactions we'll address are decomposition reactions, synthesis, and exchange reactions. Decomposition reactions are catabolic reactions, synthesis reactions are anabolic reactions, and exchange reactions can be either, okay? All right, so when we're talking about a synthesis reaction, we're talking about an endergonic or energy-consuming reaction. When we're talking about a decomposition reaction, we're talking about an exergonic or energy-producing reaction. And it's the energy produced by decomposition reactions that is used to allow synthesis reactions to take place, okay? So in a decomposition reaction, a large molecule is cracked into the smaller ones. And when that cracking occurs, energy that comes out is used to do work, okay? So an example here is the hydrolysis of a molecule of sucrose. This is table sugar. This is what you buy in the store, put in your coffee. And when we hydrolyze this, we take a water molecule and we use that to crack this connection between these two rings, and we generate now a glucose and a fructose molecule. And there's energy that comes out of that reaction, okay? We'll write energy coming out as you say En, energy, okay? Energy comes out, that should be a period, okay? And it can be used to do work, all right? The work can be, ultimately in this case, the work is going to be to generate ATP for the cell, all right? This is one of the early steps in doing that. A synthesis reaction, we take two smaller molecules, or in some cases atoms, and we connect them to each other, and the result is a larger molecule, and energy has to be consumed in order for this reaction to occur. So we'll put the energy here, En, going into the reaction on this side, okay? This is an example of a dehydration synthesis reaction where we have two building blocks, in this case they're amino acids, and we make a peptide bond between the two of them. This happens in the cell at an area called the ribosome. So peptide bond, P period, B period, right? And that consumed energy in order to occur, and this is one of the steps in building a protein, right? So the energy that's consumed, in this case, is coming from ATP. All right. So without ATP, this isn't going to happen all by itself. All right. We have to have ATP on this side, and over here we probably have the breakdown products of ATP, ADP, and phosphate. All right. ADP and phosphate. Okay. And the energy that came out came from cracking that phosphate group off of ATP, and then the energy used to build that new bond, right? In an exchange reaction, we move a chemical group from one place to another, all right? This is an example of an exchange reaction in which a phosphate from an organic molecule is attached to ADP in order to make ATP, all right? And whether or not this is an endergonic or an exergonic reaction depends on the relative energy of the molecules, okay? There is probably more net energy on this side of the equation, all right, than there is on this side of the equation. There's less energy over here, okay? And that's why creatine phosphate is able to donate to ADP, a phosphate group, okay? And that reaction isn't likely to go in the opposite direction, okay? In an oxidation reduction reaction, what happens is that electrons move from the molecule being oxidized to the molecule that's being reduced. So the molecule that's being oxidized is losing chemical potential energy, and the molecule that's being reduced is gaining chemical potential energy, okay? So a chemist would say that the oxidized group, okay, is going to have a positive change in its oxidation number, and the reduced group would have a negative change in its oxidation number. That's the formal definition of oxidation and reduction that you'll see in a chemistry textbook, right? But our definition is a little different. Oxidized means that it's losing electrons and losing energy, and reduced means that it's gaining electrons and gaining energy. And these two things always have to happen together because the electrons can't simply leave the molecule being oxidized and disappear. They have to go somewhere. An example is the conversion of NAD to NADH, right? NADH is formed when NAD picks up electrons and a proton, right? And it gets it from some other source, right? The thing that's being reduced is the NAD, and the thing that's being oxidized is whatever is donating the electrons to this molecule, okay? This happens, for instance, in the Krebs cycle. Different forms of NAD are shown here. The energy-rich molecule glucose can be used to produce NAD by giving up electrons and a proton, okay? And it has to do that in two rounds. And we generate two molecules of NADH from two molecules NAD, two sets of electrons and two protons, and the result is a high energy NADH molecule. The way to determine whether or not a molecule is oxidized or reduced compared to another one is to simply count the number of carbon-hydrogen bonds in the molecule. Okay, so if we look here, the ribose, the phosphates, the second ribose, and the adenine, those don't change between this side of the reaction and this side of the reaction. So those are the same. So the only hydrogens that we need to count are the ones that are attached to the functional group, and I'm going to go ahead and draw those in. Let's see here. Yeah, I'll draw those in for you so it's not confusing. Anytime you see a drawing like this, the hydrogens are implied, but I'm going to draw them in so that you don't have to guess where they are. Okay? This is a six-membered ring that has what's called conjugated double bonds in it, right? And that means there's a double bond, then a single bond, then a double bond. So this whole structure here is flat like a frisbee. And the number of carbon-hydrogen bonds we can count, right? 1, 2, 3, 4 carbon-hydrogen bonds, right? And then when we add 2 hydrogen atoms and 2 protons with their electrons, that's what a hydrogen atom is. Look how this changes, okay? Let's put the implied hydrogens in so you can see them, okay? Okay, and we'll count the number of carbon-hydrogen bonds here in order to determine the energy content. 1, 2, 3, 4, 5, right? Whereas over here you had 1, 2, 3, 4, all right? So this has more energy because it's got more carbon-hydrogen bonds, okay? More energy, and so it's more reduced. So this is a quick way of determining the energy content of a molecule, is just to go through and count the number of carbon-hydrogen bonds, because that's the highest energy bond for living systems. An exergonic reaction is just one that gives off energy as it goes from left to right, from reactants to products, and an endergonic reaction is one that consumes energy as it goes from left to right, from reactants to products. So endergonic reactions are generally anabolic, and exergonic reactions are generally catabolic, okay? You can see the difference between the two, right? In an exergonic reaction, the reactants have a lot of energy associated with them, and at the end of the reaction, the products have very little, right? And so energy got released from going from reactants to products, whereas over here, reactants have very little energy, and energy gets supplied in order to get to the energy content of the product. So where does this energy come from, right? And it comes from good old ATP, right? Which is the energy available in living systems to do work, right? So what you're getting here is a donation of energy in order to make this uphill climb, right? A good example would be your car driving uphill, right? If you just had an empty tank, right, and you parked your car at the bottom of the hill and you release the brake, right, your car is not going to roll up the hill. Y'all know that, right? But you can drive your car up the hill by doing what? Putting gas in the tank, turning the ignition key, then pressing on the accelerator pedal. What happens then is that the gas in the tank goes in to the carburetor, right? The carburetor mixes it with air. It then sprays it into the engine block where the pistons are. A spark plug makes a spark, and then there's an explosion inside the engine block, and that makes the pistons go up and down, and that turns the camshaft, and that eventually turns the wheel of the car, and your car goes uphill, right? So your car did something going uphill that it couldn't do on its own. It had to borrow energy from where? From the breaking of chemical bonds in the gasoline in the gas tank, right? So when you crack that chemical bond and you release that energy in the form of heat and pressure and noise, that energy got captured and used to turn the wheels of your car, and up the hill you went, okay? In living systems, we have continuous cycling of ATP, where ATP is used to do work. The phosphate group is stripped off of ATP to make ADP, and then we have to take those products and we have to put them back together to make ATP again. How do we do that? Well, we have to use energy from other sources, which includes the fuel that cells use to take in and power the activity of life, right? So that comes from carbohydrates and our lipids, and to a lesser extent, unless we're in extreme circumstances, our amino acids and our proteins, right? So the way that this works is that ATP is going to be oxidized in order for endergonic reactions to happen, and it's going to be formed when we release energy from a fuel molecule and use that to reduce ADP and phosphate to ATP. Okay, the cell can't store a large amount of ATP inside itself because that would result in an impractical amount of physical space being donated to the storage of ATP, because ATP would have to be hydrated. It would have to be solvated in water, and that would take a lot of room inside the cell, in addition to the fact that ATP is unstable. ATP likes to hydrolyze and release that phosphate group even if it's not being used to do work. Okay, so it's a very fragile temporary form of energy storage, which is why we only keep a little bit of it in the cell at any one time. So as we burn ATP, right, and release energy to do work, we regenerate these breakdown products, and then the energy from our fuel is used to regenerate the ATP, and we keep cycling as the energy needs are met, right? We talked earlier about the fact that chemical reactions are written with the reactants on the left and the product on the right. An irreversible reaction is one in which essentially there's only one direction that the reaction can go. Theoretically, all chemical reactions are reversible, but in reality, they're not, primarily because of energy differences between the reactants and the products, or in some cases, the fact that products are continuously drained off of one end, or reactants are continuously added to the other end. Okay? So, in a reversible reaction, we have a much lower energy difference between the reactants and the products, and what happens at equilibrium is that you'll have a certain ratio of reactants to products, right? So in order to make the equilibrium ratio remain constant, right, if we increase the reactants or we decrease the products, right, that should be a decrease, or if we decrease the reaction or we increase the products, that should be an increase, all right, you can see what will happen, right? We increase the reactants, we add more A and B, we upset this ratio, what's going to happen? We're going to make more of the product in order to get the ratio back where it was. If we drain off the reactants, okay, or we increase the products, all right, then, or sorry, if we drain off the reactants or we increase the products, then we're going to go in the other direction in order, again, to maintain that equilibrium. Okay, an equilibrium is a characteristic that goes with a chemical reaction, just like your fingerprints are a characteristic of the digits of your hand, right? The reaction will always try and set itself to a particular equilibrium, all right? The reaction rate is just the speed with which a chemical reaction happens. The activation energy is what has to be overcome in order for the reaction to take place. In order to overcome the activation energy in a chemical reaction, we can increase the concentration of the reactants, or we can warm up the chemical reaction, or in the event that either one of those isn't going to work, we can add something called a catalyst, okay? And a catalyst is simply a third molecule that takes place or participates in the chemical reaction without being destroyed by it. And all it does is it helps the collisions that produce the product happen, all right? So it comes into the chemical reaction, it grabs the reactants, it forces them to produce the product successfully with every collision, then it moves on to the next set of reactants. And so it doesn't take very much of a catalyst to speed up a chemical reaction significantly. Okay? Now, what we have to consider in a living system is that we can't really play around much with temperature and with an increase in the concentration of products or a decrease in the concentration of reactants without changing conditions in the cell to the point where the cell gets sick and dies. And that's why, in order to make products quickly enough for the cell to be able to use them, we use catalysts in order to help this happen. And most catalysts in living systems are proteins that are called enzymes. And enzymes will catalyze particular chemical reactions by binding to what's known as their substrates. So just like a key fits into a particular lock, an enzyme will only attach to a particular set of substrates and make one particular chemical reaction happen. Okay? An uncatalyzed chemical reaction is one that would happen without any catalyst present, and a catalyzed one will happen with a catalyst present. So in living systems, that would mean the enzyme is present in a catalyzed reaction and not present in an uncatalyzed one. Okay? The idea is to speed up the rate of the chemical reaction to the point where we don't have to wait very long in order for the reaction to happen. All right? So what I want to do here is sort of show you a little bit about that. So let's take a peek at chemical reactions.
For any reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy?
Heck on it.
For any reaction to occur,
Hold on.
For any reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy? Reactions can occur quickly and precisely. A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme's substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, Each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of the cell's metabolic processes.
So you can see here that you've got the ability with a catalyst to speed the reaction up without creating conditions that are toxic to the cell. And so you've got thousands of enzymes catalyzing thousands of chemical reactions. And the unique thing about enzymes is not only do they have secondary, tertiary, and sometimes quaternary structure that makes them perfect for their job, they also have the ability in many cases to be regulated so that once they generate enough of a particular product for the cell to use, they can then be turned off or turned down so that we don't continue to generate something that's no longer necessary for the cell to function. The active site on the surface of an enzyme is a cleft or a pocket in which the substrate can fit. Many enzymes will then change shape. This is known as induced fit, in order to aid the chemical reaction that it's trying to catalyze, and then will release the product and then resume on a new set of reactants. Enzymes can be found almost anywhere. They can be found in the nucleus of the cell, they can be found in the cytoplasm, on the rough endoplasmic reticulum, inside cellular organelles, on the surface of cell membranes, or in some cases on the surface of cellular organelles. All right. Some are secreted from the cell into the external environment, and some remain embedded in organelle membranes or in plasma membranes. Substrates generally enter the active site and form a complex, whereupon the enzyme will change its shape in order to catalyze the breakdown of that particular reactant. And then the products get released and the enzyme can repeat the process just like is shown here, right? So you can see here how we can bind, right? We can strain the chemical bond from induced fit, cause it to break, release the products, and then cycle back and do the same thing again, okay? In a synthesis reaction, we bind the reactants, we utilize energy to form a novel chemical bond, we release the product, and then we can go around and repeat that process again, adding another link chain of the building of this particular polymer, okay? This would be an example, for instance, of glycogen synthase, right, which makes glycogen from glucose, hooking glucose molecules together. Occasionally, enzymes require help in order to do their work, and that can come in the form of a cofactor or a coenzyme. A cofactor is generally a mineral ion that sits in the active site in order to help the enzyme do its work, and we get cofactors from our diet, while a coenzyme is generally a vitamin or a modified vitamin that helps the enzyme do its work. And then sometimes you'll have enzymes that have both cofactors and coenzymes in them, okay? Some enzymes don't need cofactors or coenzymes. It all depends on the type of enzyme we're talking about. You can see some of the different enzyme categories here: oxoreductases, transferases, hydrolases, isomerases, ligases, and lyases. Transferases catalyze the transfer of a chemical group from one molecule to another. Oxoreductase will oxidize one molecule and reduce another one. A hydrolase will use water to split a bond. An isomerase will change the order in which atoms are connected in a molecule. A ligase will attach molecules to a chain, a growing chain, and a lyase can be used to split a double bond, okay? So they're always named based on what they do, and they always end in ASE, okay? And from that, you can determine what it is that they do. And 99 times out of 100, these are going to be proteins, which are enzymes, right? There are some examples in living systems of enzymes that are RNA molecules, okay? But we're not going to discuss those here. The rate of a chemical reaction we've discussed, right? We've talked about the fact that we can accelerate a chemical reaction by heating it up or increasing the concentration of the reactants, but the main way that we speed it up is by adding a catalyst, okay? Note that the speed of a chemical reaction will reach a peak called saturation, at which all the enzymes catalyzing a potential chemical reaction are working at their maximum speed because they're all binding their substrate molecules. And beyond that, adding more substrate will not speed up the chemical reaction. Okay? The same is true of transport proteins that work on this principle. They have a saturation point as well. So a maximum speed can be attained. Temperature, we know, is going to speed up chemical reactions, but there are always going to be optimum temperatures under which every enzyme is going to operate. Okay? These optimal temperatures are the temperatures at which the enzyme can maintain its normal shape and perform its activity the fastest. All right? When you're below the optimum temperature, collisions between the enzyme and the substrate happen too slowly, and when you're above the optimum temperature, the enzyme begins to denature, and eventually it'll no longer function. Okay? So in most enzymes in the human body, the optimum temperature happens to be body temperature. That's very convenient, right? The same can be said for an optimum pH and an optimum salt concentration, okay? There's going to be an optimum pH at which enzymes will work, and that pH in enzymes in the human body is generally between 7.3 and 7.45. There are some areas in the body that have different pHs, and the enzymes that work there have different optimums. One example would be the stomach, which has a pH of around 2, and the enzymes that operate in the stomach do their best work around pH 2. Okay? If you're above or below the optimum pH, what happens is that the secondary and tertiary structure break down, and the result is that the enzyme doesn't function as well. Now, we can control enzymes using inhibitors and enhancers. Okay? An inhibitor is a molecule that binds the enzyme either within the active site or at what's called an allosteric site in order to slow it down. Okay? A competitive inhibitor is one that will bind to the active site but is not capable of producing a product, and its effects can be overcome with increasing concentration of substrate. An allosteric inhibitor doesn't bind at the active site. It binds at some other site, and it changes the shape of the enzyme, and it cannot be overcome with increasing amounts of substrate. Okay? And in the body, there are examples of both competitive and non-competitive inhibitors that can control the speed at which an enzyme works. Generally, the inhibitor is the end product of a metabolic pathway that, once it builds up to a particular concentration, will bind to the allosteric site if it's a non-competitive inhibitor, or bind to the active site if it's a competitive one, and that slows down the entire metabolic pathway. Okay? So the idea is that once you get to a certain concentration of a product that the cell wants, we don't need to keep making it, because that would be a waste of time and resources. So it builds up to a particular concentration, it binds to the allosteric site or the active site, depending on how that enzyme is controlled, and the entire pathway either slows down or it stops, right? So there's an example with a competitive inhibitor, and there's one with a non-competitive inhibitor, okay? You can do the same with enhancers. There are examples of metabolic pathways that, once a particular molecule builds up to a specific concentration, it speeds up a particular metabolic pathway so that you can make more of a particular product more quickly. And there are activators that are allosteric, and there are some activators that can bind in the active site. Okay? So a metabolic pathway is simply a collection of chemical reactions that generate a final product, all right? Metabolic pathways can run in a straight line, they can run in a circle, they can fork, they can fuse, okay? It all depends on the type of metabolic pathway you're talking about. Generally, with a metabolic pathway, the enzymes that participate in it are in close proximity to each other. So they may be attached to protein scaffolding inside an organelle or cytoskeletal components in the plasma, the, the plasma membrane or in the cytoplasm of the cell, okay? And that allows them to be more effective at their job because we don't spend as much time worrying about random collisions, right? The product of one catalyzed reaction becomes the reactant of the next catalyzed reaction. So those are the advantages of multi-enzyme complexes. And that's just a statement of how negative feedback works, right? We generate a certain amount of product at the end of a particular metabolic pathway, and we don't need to keep cranking that pathway through, so we can turn it down or turn it off until concentrations of that product fall to the point where we need to turn the pathway back on again. So you can see examples here of how that works, right? Here's enzyme 1, 2, 3, and 4, a straightforward pathway. The initial substrate becomes a product of this enzyme and a reactant for this enzyme. And the same happens here and happens here. And then eventually we get to the end product. And once it builds up to a certain concentration, we shut down or shut off an earlier enzyme in the pathway, and the whole pathway then slows down, okay? And that makes us more efficient. Enzymes can be regulated by phosphorylating them. The phosphorylation can either activate or inactivate them. The enzymes that phosphorylate other enzymes are called protein kinases. And then they can be dephosphorylated by phosphatases, which can turn on some enzymes and turn off other enzymes, okay? An example of drugs that act as enzyme inhibitors include things like penicillin, which can target a bacterial enzyme that helps to build the cell wall of certain bacteria. And the result is that the cell wall is incomplete. And when the bacteria hit an osmotic challenge, they rupture and die, okay? Viagra inhibits phosphodiesterase, which breaks down cyclic AMP in the cell. Cyclic AMP is known as a second messenger, so it's a molecule that's produced after a series of interactions of enzymes and substrates that will initiate certain responses within the cell. Well, in Viagra, what happens is that the phosphodiesterase that breaks down cyclic AMP is inhibited. The cyclic AMP hangs around longer in the smooth muscle around the blood vessels that, when they dilate, bring more blood into the penis, and the result is that the penis stays erect for a longer period of time, okay? Viagra was actually first marketed as a hypertension medication, and it didn't do anything for blood pressure, but it turned out that all the male participants in the study ended up with erections, and so it was remarketed as an ED cure, right? And so that's its current use today. It also happens to be the same pathway that Rogaine took, which is the drug that can regrow hair on bald male heads. Lactose intolerance is an example of a condition brought about by a reduction in the production of lactase, which is an enzyme that breaks down the disaccharide lactose into galactose and glucose. And in individuals that have lactose intolerance, the reduction in the amount of that enzyme means that lactose in the diet ends up making it all the way to the large intestine, where the E. coli and other bacteria that live there use it for fuel, overgrow, and produce gas, distension, and pain. And so to deal with this condition, people who are lactose intolerant can take the enzyme in pill form or they can avoid lactose by having dairy products that are lactose free. Now, cellular respiration is a series of chemical reactions that generate the ATP needed by the cell in order for the cell to function. And it's made up of three big legs, okay? Glycolysis, the Krebs cycle, and the electron transport chain. The overall chemical reaction for all of those pathways is shown below, okay? And as you can see down here, okay, the equation is balanced, right? If we look here, there are 6 carbons on this side. 6 times 1 is 6 carbons on that side. There's 12 hydrogens over here. There are 6 times 2, 12 hydrogens over there. There are 6 oxygens plus 12 oxygens, which is 18 oxygens on this side. And there are 6 times 2, which is 12 oxygens here, plus 6, which is 18 oxygens over here. So this is a balanced equation, okay? The pathways for ATP production, again, glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis happens in the cytoplasm. It starts with glucose and it ends with the production of pyruvate. The Krebs cycle happens in the mitochondrial matrix, which is in the core of the mitochondria, and it begins with pyruvate being converted to acetyl-CoA, which is added to oxaloacetate to form citric acid, which is one of the other names of the cycle, the citric acid cycle. And it ends when it cycles all the way back to oxaloacetate, waiting to get another acetyl-CoA group, okay? And it produces ATP directly, FADH2 directly, and NADH directly, okay, as well as kicking out carbon dioxide. The electron transport chain accepts NADH and FADH2. It generates water, and it generates heat, and it generates most of the ATP generated by the 3 legs of metabolism, and it takes place in the intermitochondrial membrane, okay? The 2 types of energy production that happen in metabolism are substrate level phosphorylation of ADP to make ATP, and oxidative phosphorylation, where we first transfer high energy electrons to carriers like NAD and FAD, and then we oxidize them in the electron transport chain and use that power of those electrons moving down the chain and giving up their energy to generate a proton gradient in the intermembrane space of the mitochondria, which is ultimately released through ATP synthase, where most of the ATP gets produced, okay? There are 20 different enzymes required for all 3 legs of metabolism, and they're found in the cytosol and the mitochondria. And there's the layout, right? There are the 3 legs, right? The intermediate stage is really just the movement of the pyruvate from the cytoplasm into the mitochondrial matrix. So some consider that a separate phase of metabolism. Some just consider that the transition from glycolysis to the citric acid cycle. Remember that these 2 parts, citric acid and electron transport, happen in the mitochondria. Glycolysis is out here in the cytoplasm, okay? Glycolysis is the only part of these 3 legs that does not require oxygen to produce significant amounts of energy, okay? So it can happen in both an anaerobic and an aerobic state. It happens in an aerobic state. It ends in pyruvate, produces NADH, produces some ATP, and is a fairly rapid pathway, right? And then that pyruvate ends up going into the citric acid cycle. If the oxygen levels are too low, then what happens is that it goes into what's called a fermentation cycle, in which the end product pyruvate gets reduced to lactic acid, and then the cycle gets repeated, and the lactic acid is actually removed, put into the bloodstream, and then sent to the liver, okay? And then once those low oxygen conditions are reversed, the liver will send the lactate as pyruvate back to the muscle, and the muscle can then use the pyruvate to generate more ATP, right, in order to restore its energy reserves, right? You can make 20 times more energy in the presence of oxygen than you can without it, all right? Glycolysis doesn't require oxygen. There are 10 enzymes in the cytosol that participate in this. The end products are 2 pyruvate molecules, 2 ATP molecules, and 2 NADH molecules, okay? So let's take an overview of glycolysis, and then I'll talk a little bit about each step in some more detail. So we'll bump out of this. And we'll look at glycolysis.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid. Cells derive energy from the
So this is an extremely rapid pathway, okay? Very, very quick. In the first 5 steps, we split glucose into glyceraldehyde 3-phosphate, and we actually use some ATP in order to trap the glucose inside the cell so that it doesn't leave the cell through a leak channel. And we generate glyceraldehyde 3-phosphate, okay? In steps 6 through 7, phosphate is added to substrate and 2 hydrogen atoms are released to NAD, and we generate some NADH, and phosphate is transferred from ADP to ATP. Okay, so you can see here that we're generating a couple of NADHs, right? And that we're generating a couple of ATPs here, all right? And then step 8, molecule from step 7 gets converted to an isomer that doesn't require any energy. And then we lose a water molecule, and then a phosphate gets transferred to form another ATP molecule, all right? So what will happen here is that we generate through enolase something called phosphoenol pyruvate, or PEP. And this phosphate bond is so packed with energy that it can be used to send a phosphate to ADP, making ATP, all right, because the energy content here is actually greater than the energy content of the ATP, and then we generate now pyruvate, which is eventually going to move into the mitochondria, okay? This is a rapid pathway, but it doesn't yield very much energy. If we don't have oxygen around, all right, we're going to end up with a net production of 2 ATP for every glucose that we burn. Now, we can do this very quickly, but under extreme high energy consumption circumstances, this isn't going to sustain activity for a very long time. And eventually what will happen is that you'll run out of energy and you'll have to stop the activity, right? And at that point, you're going to pay your oxygen debt by breathing hard, and then you're going to restore your energy stores and your glycogen stores, and then you'll be able to eventually restart the activity that you stopped, okay? We talked about the fact that in high oxygen situations, you can run glycolysis all the way to pyruvate and not worry about running out of energy, because the demand is either equal to or lower than the ability of these chemical reactions to supply the energy, all right? But if the oxygen levels are low, because the energy demand is higher than the rate at which these chemical reactions can supply the energy, then what's going to happen is you're going to go into lactic acid metabolism. And the reason for doing that is to regenerate NAD in order to continue to run glycolysis. But eventually what happens is that the energy levels get so low that the activity is going to stop, all right? Next thing that's going to happen, we're going to move the pyruvate into the mitochondria, forming something called acetyl-CoA. So what will happen is that a carbon dioxide will be stripped off of pyruvate, and the acetyl group will be added to coenzyme A, and the energy from decarboxylation is so significant that we can use it to form an NADH from an NAD, okay? So that is a big time energy yielding step, and all that NADH and FADH2 is eventually going to pay off when we get to the electron transport end. So this will occur twice. 2 NADH will be produced, okay, and then we'll be ready to begin the citric acid cycle, the Krebs cycle, okay. So that is our next phase. So let's look at the Krebs cycle, okay. So we'll go to here. Okay. Come on. There we go.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2.
These reactions regenerate the 4-carbon molecule that initially reacts with acetyl CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
So this cycle
During glycolysis,
can run.
glucose is broken down to pyruvate.
This cycle can run without oxygen and produce a small amount of ATP, right? But eventually it's going to run out of intermediate molecules and stop, okay? So this isn't going to do us much good in a low oxygen situation, in addition to the fact that it's not going to be receiving any pyruvate from glycolysis, okay? So this will shut down pretty quickly. Again, the end products, right? You're going to break, for each crank of the cycle, 3 NADH, 1 FADH2, and 1 ATP. So for every glucose molecule, you're going to double that up. So 2 ATP, 6 NADH, and 2 FADH2, right? So those are the steps, right? Acetyl-CoA combines with oxaloacetate to form citric acid. We isomerize that. We transfer hydrogen to NAD to form NADH, and we generate a CoA. We remove the CoA and form ATP. Dehydrogenase transfers hydrogens to FAD to form FADH2. We take a water off, and the dehydrogenase transfers hydrogen to form NAD. Those are the individual steps, right? There's the formation of, let me get the laser pointer out here. There's the formation of citric acid, acetyl-CoA, and oxaloacetate. So that's one of the ways that we get the name for the cycle. The citrate gets isomerized to isocitrate. And then we strip off a CO2. That's an energy yielding event, and we can use that to make an NADH. We form alpha-ketoglutarate. We add a CoA and strip off a CO2 and make another NADH. So every time a CO2 leaves, an NADH gets formed. Then we strip off the CoA. That allows us enough energy to make an ATP. The succinate gets oxidized. The FAD gets reduced. We generate fumarate. We add a water molecule to form malate. We now are going to reduce an NAD to make NADH, and oxaloacetate reappears at the top of the cycle. Okay. Now, when you go into ketoacidosis, all right, what happens is that you eat a diet that's low in carbohydrates, which means you run low on all of these guys, right? So you run low on malate, fumarate, succinate, succinyl-CoA, oxaloacetate, and so on. And so what happens then when you're eating nothing but, say, fat and protein, is that all you have is acetyl-CoA from those steps. You can't make much of these guys. And so what happens is that the acetyl-CoA ends up, instead of forming citric acid, it ends up forming what are called ketone bodies. And where do the ketone bodies go? They go out of your body in your pee, in your sweat, your breath, and you literally vaporize your fat. And you start to lose lots of fat weight. That's good. What's bad is that because these keto acids are acids, they lower your body pH to the point where you can get very sick and even die. All right. So this is what they try to do in South Beach and Jenny Craig and all these keto diets that you see out here. All right. In addition to this, you run the risk of having such a low fiber content in your diet that you can suffer from extreme constipation. All right. So it's kind of a dangerous road to go unless you're under a doctor's supervision. Okay. The citric acid cycle occurs in the mitochondria. It needs oxygen to really run for an extended period of time. The initial substrate is acetyl-CoA. Every crank of the cycle gives you an ATP, 3 NADHs, and 1 FADH2. right? And 2 turns is going to give you what's shown below, all right? So notice that up to this point, we haven't really made a lot of ATP, all right? We've made, 4, which is, it's okay, but that's really not going to last you too long in intense activity, all right? So where do we get all the rest of the ATP, right? We're going to end up making about 40 of them. Well, that occurs in the electron transport chain, right? The way we regulate the citric acid cycle is that if our energy levels are high, the cycle slows down, and if our energy levels are low, the cycle is going to crank up, all right? And that's the result of, again, feedback inhibition and also probably some feedback activation. The electron transport chain is the last of the 3 legs, all right? And it's where we get the big payoff. That's where we get the most energy from the food that we eat. So let's take a look at how the electron transport chain works. Okay.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD plus and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, ions from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD plus and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by mechanism is referred to as oxidative phosphorylation.
So it's the setup of this proton gradient across the intermitochondrial membrane that ultimately drives ATP synthesis, because it gets relieved as it flows through something called ATP synthase. That's the only place where that proton gradient can relieve itself, right? The oxidation of the NADH and the FADH2 make NAD and FAD, and then they go back to the electron transport chain to be reduced again, and that cycle repeats, okay? So what happens is, as these electrons move through the chain, is that the energy captured by that movement, as the electrons change their location, is used to pump protons from the matrix into the intermembrane space. And that ultimately is going to drive ATP production, okay? It turns out that the energy content in an NADH is approximately enough to make 3 ATP molecules from ATP synthase, while that from FADH2 is about enough to make 2, okay? Oxygen is the final electron acceptor in the chain. When the low energy electrons get there and the protons attach, you form water, and then the water drifts away, and then the chain can continue to flow, okay? If anything other than oxygen is at the end of the chain will constipate and will stop flowing, and the proton pumping activity will stop, and the result is that ATP synthesis will stop, all right? This happens, for instance, in the presence of carbon monoxide or cyanide, because they have about the same shape as an oxygen molecule, and that's why they are metabolic poisons, okay? This generates 32 of the 38 ATP that we use, or that we make, by completely oxidizing a molecule of glucose using glycolysis, the intermediate stage, the Krebs cycle, and the electron transport chain, okay? So as long as electrons flow, we continue to crank out the ATP, all right? There are compounds that can interrupt this flow. An example is a compound called dinitrophenol, which can cause the intermitochondrial membrane to become leaky and the protons diffuse into the matrix without going through ATP synthase. Dinitrophenol used to be used as a diet aid long ago. People would take it and they would lose a lot of weight. The problem is it's a poison, and if they took too much, they could die, okay? In addition, there's a molecule called rotenone, and what rotenone does is it sits on top of some of the ATP synthesis apparatus and stops the production of ATP, all right? So all of these are very dangerous poisons to be exposed to, okay? The number of ATP molecules generated depends on where the electron donor enters the chain. We talked about the amount of power in an NADH versus an FADH2, okay? And we talked about ATP and glucose breakdown, okay? If we do the math, all right, then we end up with an estimate between 34 and 38 ATP, depending on the textbook you read and really depending on the individual, okay? The reason that ATP production can be different from person to person, all right, is because there are different flavors of the enzymes that do the work in each of the 3 legs of metabolism, and some of those flavors of enzymes are less efficient than others, and as a result, the ATP production will be different, right? The less efficient the enzyme, the more likely we're going to lose more work as heat than trap it as ATP, and those people tend to be skinny, right? And the more efficient those enzymes are, the reverse tends to be true. We get more towards the 38 ATP end, and the tendency there is that we'll pack on weight, okay? So one of the ways to control your weight is to try and change your metabolism in a non-genetic fashion, and that's by doing more exercise and generating more tissue in your body that has more mitochondria in it, and as a result can burn more fat even when you're resting, to make ATP to do the work of building more muscle. All right, and when you do that, then you end up in what we call a calorie deficit, more likely than not, and you'll tend to lose weight. Okay, so we talked a little bit about cyanide poisoning. One way to know if you're around cyanide is it smells and tastes like bitter almonds. Okay, carbon monoxide is different. Carbon monoxide is odorless, tasteless, and colorless, and that's why it's deadly. Okay, the only way to know that you're around carbon monoxide is with a detector. Okay, which is why you should have one in your home with good batteries in it. You read every winter about people using kerosene heaters in poorly ventilated areas that end up going to sleep and not waking up because the fuel burns all the oxygen out of the air, creates carbon monoxide, and they end up dying from carbon monoxide poisoning. Okay, this is also one of the ways that people commit suicide is by running their car exhaust into their driver cabin in a taped up window, and after about half a tank of gas, they're dead. Okay, and that's from the carbon monoxide poisoning. We talked also about the fact that in the presence of insufficient oxygen, you're going to make lactic acid, which will leave the cell and go to the liver, and eventually the energy deficit will get to the point where the activity can't continue and you'll stop. Okay, at that point, the lactic acid will be sent back to the muscle as pyruvate, and we can then recover our energy stores and our fuel deficit, and we can then prepare ourselves for the next round of activity. Okay, so with insufficient oxygen, we regenerate the NAD to try and keep glycolysis going because the other two legs of metabolism can't run. We don't really make enough ATP for the activity to continue very long doing this, and the result is eventually we quit. We go to what's called failure. All right. Now, fatty acids and amino acids can also be fuel molecules. Fatty acids enter the Krebs cycle through what's called beta oxidation, where we start at the acid end of the fatty acid and we break off acetyl-CoA molecules. So those are two carbon units. They enter the Krebs cycle, combining with oxaloacetate to form citric acid, and then you get one crank of the cycle, right? So you go through, and a typical fatty acid is going to be about 20 carbons long. So you get 10 turns of the cycle with one fatty acid, which is why when you look at a triglyceride with 3 fatty acid tails, you're looking at a lot of ATP, right? That's the demonstration that there are more calories, more energy in a triglyceride than there are in a carbohydrate or in a protein, in an amino acid, okay? Amino acids are only used for fuel if we're in a starvation scenario, okay? Generally, the reason that you eat protein is to make more protein. That's why one of the ways to lose weight is to go high protein, high fiber, right? So amino acids undergo what's called transamination or deamination. They strip off the amino group and they become what are called alpha keto acids. And they can enter the Krebs cycle after isomerization takes place and then be used for energy. But this is only going to happen if you're not either eating or you haven't stored any lipid or any carbohydrate. Okay, so that's one of the things that you see, for instance, in people that have a low protein diet. Right, is that they have to utilize their protein in order to make body protein. They end up causing their muscles to waste. Okay, and the result is that we're just trying to make as much energy as we can on a diet that really doesn't have a lot of calories in it, or a diet that's terribly imbalanced, okay? And the result is a condition called ascites with a distended abdomen because of the fluid collection that happens there due to the fact that the blood is losing fluid to the abdomen through osmosis because the blood doesn't have enough blood protein in it to hold on to its fluid. And you also see muscle wasting in those individuals. And so the fix for that is to put protein calories back in the diet, and then they can resume a healthy biochemistry and live a longer life.